How can you make a football field this big? Hi biology friends, it's Sarah, your other biology professor, and let's talk about chromatin and DNA packaging. Because your DNA isn't just naked in the nucleus. In eukaryotes, our DNA is associated with proteins, and we call that complex of DNA and protein chromatin. One molecule of double-stranded DNA plus its associated proteins make up a chromosome. So chromosomes are made of chromatin, DNA plus protein. But why bring in the protein? We needed to package the DNA, and DNA packaging accomplishes a few important things. It's a space saver. One of your cells has about 2 meters of DNA. There's a lot of A's, C's, G's, and T's to cram into that nucleus, and packaging can compact DNA down to about 1/10,000th of its stretched out length. So why not just ball up the DNA and squish it in the nucleus? Well, DNA isn't just long, it's also negatively charged, and all those negative charges would just repel each other. You couldn't keep the structure tight. We need the proteins. The packaging is also protective. It prevents the DNA from becoming tangled and damaged during cell division when chromosomes are being pulled around. And it's an important part of regulating gene expression, controlling how much product is made from genes. Let's check out the structure of that packaging. Most of the proteins you'll find in chromatin are histones. Histones are little tailed proteins, each made of one polypeptide chain, amino acids strung together. Histones have a high percentage of 2 amino acids in particular, lysine and arginine. Here they are. What do you notice about the side chains of those two? They're positively charged, which means they'll interact nicely with the negatively charged DNA. Histones come together to form an octamer. 2 each of 4 different histone types form an octamer, a molecule made of 8 subunits. In the octamer, the histone tails stick out and can interact with the DNA to stabilize the structure. Our DNA coils almost 2 full times around each octamer, sort of like thread on a spool, wrapped to form a nucleosome. The DNA in between nucleosomes, not wound around histones, is called linker DNA. This level of packaging is often called beads on a string, with nucleosome beads and linker DNA string joining them. But you may see it less whimsically called the 10 nanometer fiber because the nucleosomes are about 10 or 11 nanometers in diameter. Nucleosomes can be reversibly modified or moved to clear a path for DNA replication or transcription proteins to move through. This first level of DNA packaging is the one we understand the best. As we move into higher levels of packaging on the way to the most compact chromatin, you'll notice 2 things. First, condensation will make the chromatin shorter and thicker. And second, we're still working out exactly what's going on. To get us to the next packaging level, a 5th type of histone, different from the 4 pairs in the nucleosome core, binds to the linker DNA in between nucleosomes. But this one isn't part of the bead. Instead, it acts like a clip, grabbing up stretches of linker DNA, adding stability to the area where DNA enters and exits the nucleosome. This structure of nucleosome with the extra histone is called a chromatosome, if you run into that term. This additional histone, a linker histone because it binds the linker DNA, is essential for further compacting the chromatin. Grabbing the linker DNA pulls the nucleosomes closer together, and the linker histone's tail neutralizes negative charges in the linker DNA, encouraging the nucleosomes to stack and fold into a structure called the 30 nanometer fiber, getting wider and shorter. The tails of the nucleosome histones interact with DNA in their own nucleosome and with adjacent nucleosomes to contribute to this compaction. The compactness of the 30 nanometer fiber makes it hard to see the exact arrangement of the nucleosomes in the stack. Different models exist to show possible arrangements, but in living cells, chromatin at this level isn't organized in the uniform fiber you see in textbooks. It's constantly changing, maybe more like clumps of nucleosomes mixed with some stretches of beads on a string, or a melty, movable arrangement. Reality is messy. Either way, the chromatin further condenses by forming loops, stabilized by non-histone scaffold proteins. And while it might seem like a loopy, thready mess, there's organization here. Loops can bring far apart regions closer together, with the interactions affecting gene expression. But when the cell divides, we have to move things around. During cell division, even more scaffold proteins help to further compact the chromatin as the loops fold onto themselves to form the most condensed and visible structure, a metaphase chromosome. These super compact chromosomes are easier to move around without damage and easier to keep track of, making sure the right number of chromosomes ends up in each new cell. After cell division, chromosomes will decondense back to their interphase state. DNA wraps around histones, forming nucleosome beads on a string that stack and compact into dynamic 30-nanometer fiber that loops and condenses into the most compact chromosomes in dividing cells. Drop me a like if you learned something about DNA packaging. Subscribe for more biology content from me. Check out my website for tutoring resources, and I will see you next time. Bye.